So we're moving on to capillaries. And just to remind ourselves of some of the features we described of capillaries, we're going to talk about their anatomy, how they differ and the different types. And then we'll talk about how they control flow to capillary beds. What are some of the additional features and structures that regulate the flow to capillary beds? And then we'll talk about how we have capillary movement, movement of fluid, movement of materials across the capillary wall. So just to remind ourselves here, capillaries are the site of exchange, so their diameter needs to be very small, about 5 to 10 micrometers in diameter. And this is a small diffusion barrier, so about 1 cell layer thick, very, very tiny distance for particles to diffuse across. They're also going to have the greatest total cross-sectional area, meaning that they are the most abundant vessel. So we have the most amount of capillaries in our entire system, and they have the greatest cross-sectional area, very large surface area. And then another feature is that this is the slowest area for blood flow. And the reason for that is to allow for exchange. So this takes time. In order for materials to move across in both directions, we want to have very slow flow across the capillaries. Now we have about 10 to 40 billion per body, so many, many capillaries, again, the most abundant. Total surface area about 600 meters square. So you're talking about larger than, a basketball court, larger than a basketball court or NBA basketball court. And then when you think about the location of capillary beds, most of our cells in our body, most of our tissues are within 1 millimeter of a capillary. So most of our tissues need to have close connection to a capillary bed in order to receive their nutrition and to, eliminate their metabolites. And then the 1 millimeter is about the length of a capillary, so not very long in terms of distance, but very short in terms of how far blood moves across this. But this is why it has to be slow, right? Very slow flow as it moves across that 1 millimeter length. Now, most capillaries are going to have pores between the cells, right? Remember, capillaries being 1 cell layer thick, they're pretty much a layer of endothelial cells stacked against each other. And so the pores in between those endothelial cells allow for a plasma to move across. This is what allows us to have that diffusion and movement of particles. However, this is going to be protein-free plasma, okay? Proteins we described previously in MP1, proteins are too large to pass through these types of pores. And so they usually need some type of, transporter or some type of channel or pore in order to move through the cell membrane. And then looking at capillary beds, these are more extensive networks of capillaries, so much more expansive capillaries that are linked up together to create a bed that will usually feed specific organs or organ systems or structures. So capillary beds are much more complex networks. All right, so just to remind ourselves here, capillaries have the greatest cross-sectional area and the slowest flow velocity, so the slowest movement of flow. And so looking at the cross-sectional area, arteries like the aorta, very, very small. Arteries, slightly higher. Arterioles, we have a larger number of arterioles, but then capillaries, about 3,000 capillaries, 3,000 being the total cross-sectional area of the capillaries, meaning that it's the most abundant vessel. And then we see the reverse trend for veins, right? Smaller number of venules and even smaller number of veins. And then looking at the flow velocity down here, we start out with flow moving through the aorta quite high. High flow going through the aorta, slightly slower in arteries, and then the flow drops off significantly in arterioles. This is because of the resistance. So we talked about this before. The greatest resistance is introduced in the arterioles, which is what slows down the flow. And then the lowest flow rate is going to be at the bottom here, which is our capillaries, and then the reverse for venules and veins. All righty. Now, let's talk about the different types of capillaries based upon their structure and the types of transport that they allow. So we have two major types. These are much more abundant than the third type. There is continuous capillaries, more common, and there are small gaps in between the endothelial cells of continuous capillaries. So very tiny pores, as we described earlier, that allow small substances to pass through. These have to be water soluble or lipophilic in order to move through these pores. And then we have fenestrated capillaries. These have larger gaps. So they do also have openings, but these are slightly larger, forming larger windows or fenestrations, and these do allow for proteins, and in some cases, larger blood cells to move through these spaces. So the openings here in fenestrated capillaries allow for larger particles to pass through. And then discontinuous capillaries are having even larger channels. This is why they're referred to as being discontinuous or sometimes sinusoids, because they have much larger gaps. They also have an interrupted basement membrane. So the basement membrane is not continuous, and it even more so allows sacs or spaces in between these cells. Okay, so continuous, fenestrated, much more common, much more important, and then discontinuous or sinusoids being the third type. Okay, now we want to think about structurally how these differ. So let's draw this out here. How do these capillary types differ in structure? And then what are some examples throughout the body? Okay, so we are looking at the anatomy of capillaries. Anatomy of capillaries. Let's look at the three types. So we have continuous, fenestrated, and then discontinuous, also called sinusoids. Sinusoids. So continuous will have a continuous basement membrane, hence the name continuous. And then it will have endothelial cells internally, one cell layer thick with really small, tiny pores in between. So this is the structure of our continuous capillary. Let's talk about some of the features here. So it's a very narrow endothelial cell gap, right? So very, very tiny gap in between here. So narrow endothelial cell gap. A continuous basement membrane, right? So continuous. And basement membrane is just the underlying connective tissue. It's what helps to nourish the cells, right? So it's not a cell layer, just that supporting tissue, but it's continuous. This is going to allow water-soluble molecules to pass through. And then the type of movement that we hear is slower, so slow movement, right? Slow movement as molecules pass through those spaces. We also want to remind ourselves this is the most abundant, so the most common. And so we're going to find this in many, many places throughout the body. It's the most abundant, so I won't list any specific examples. Okay. Let's talk about the fenestrated. Fenestrated are going to have larger fenestrations or windows. So also having a continuous basement membrane and then much larger fenestrations in between. And much larger spaces. So you can already see larger particles passing through, right? Proteins and so forth. So because we have a larger endothelial cell gap here, it's going to be the first feature, wider endothelial cell gap. We also, and we can refer to these as fenestrations. Okay. We also have rapid flow happening here just because the spaces are larger. So rapid flow, and then we allow larger molecules to pass through here. So larger molecules, for example, proteins or blood cells. Okay, so larger molecules. And then examples of this would be certain parts of the kidney where we have filtration. So the kidney, certain parts of the GI tract or the gut, GI tract, as well as most of our endocrine glands. This is how proteins leave our blood vessels and go out into the blood from our endocrine glands. So this is really important in endocrine hormone transport, so endocrine glands. Endocrine glands. Okay, this is how thyroid hormone gets released. Um, LH, FSH, most of our endocrine hormones have to be able to pass through these fenestrations. Okay, so kidney, GI tract, endocrine. Now, discontinuous sinusoids, these are going to be much different. They have a discontinuous basement membrane. So even the basement membrane itself has openings, and the openings are even larger. And then the endothelial cells are separated as well. Now, we may see the discontinuous sinusoids being in areas where we have a transition from fenestrated to sinusoid. So there can be a bit of a transition in terms of an area where we have fenestrated capillaries, there's discontinuous and then there becomes bigger sinusoids. And sinusoids are bigger channels or spaces, right? Channels or spaces. So there's kind of, again, this gradual transition here, right? So areas of transition from fenestrated to sinusoids. Okay. We're going to see these in some areas where we have blood-filled sacs, like the liver, right? Blood-filled sacs or sinusoids. And then here we're going to find free flow of material. So we have free open flow of material. And examples of this would be the liver, where we have those liver sinusoids. We also find this in the bone marrow. Again, blood-filled sacs here, as well as the spleen, right? So these hematopoietic related organs are typically where we find these dilated blood sacs, which are sinusoids, and then discontinuous is a transition. Okay, so these are the least abundant or least common and very specific examples. Alrighty. Okay, so these are examples of our different types of capillaries. Please think about how they differ structurally, how they differ functionally in terms of what can pass through there, and then the examples throughout the body, right? As well as the rate of flow. So slowest flow in continuous capillaries, rapid flow in fenestrated, and then even faster free open flow in our discontinuous and sinusoids. Okay. All right, any questions on this? Any questions? Okay, so let's keep. Okay, let's keep going. So let's talk about the local control of blood flow through capillary beds. So there are three structures we want to think about that help to regulate the flow. And there are smooth muscle modifications within the microcirculation. So first we have the arterioles, then we have meta-arterioles, which are smaller connections between capillary beds, and then we have pre-capillary sphincters. So we'll talk about the role of each of those structures, where they're found, and then how exactly they regulate flow. First off, meta-arterioles. And meta-arterioles are these smaller connecting branches in between an arteriole and the capillary bed. And it's a way for us to functionally shunt or bypass the capillary bed and correct an artery directly to a venule. So notice that the middle structure here, which is the meta-arteriole, it's going to bypass the smaller capillary structures and go from an arteriole directly here to a venule. The way that this modification happens, the way that this regulation happens, is because of the smooth muscle rings that are at the base of the meta-arteriole. So we have some here, and then we have some at the openings to the capillary bed as well, which are the precapillary sphincters. So these rings around the capillary and at the base of the meta-arteriole, along with the rings that are actually found within the arteriole itself, strategic locations of these smooth muscle bands is what helps to direct the flow either to the capillary bed or bypassing the capillary bed and taking it towards the venule. And so based upon local factors, some of which we described earlier, such as oxygen levels, CO2 levels, potassium levels, that will determine whether or not we want to have slower flow go into the capillary bed or whether we want to bypass that If the tissue bed is well oxygenated and has little CO2, we're going to bypass that, shunt blood towards the meta-arteriole and then towards the venule, and then essentially bypassing the capillary bed. The second structure here are precapillary sphincters, and these are created by rings of smooth muscle at the base of the capillary bed. So again, more strategic location of these bands of smooth muscle at the entrance to the capillary itself. So contraction here is going to be in response to local factors only. Okay, so only in response to local factors. And so, for example, contraction is going to constrict the rate of flow into the capillary bed, thereby decreasing how much blood is actually going to the capillary and shunting that blood back towards the meta-arteriole or even in the arteriole itself. And then relaxation on the other end is going to do the opposite. It's going to increase how much flow is going to the capillary bed and therefore bypassing the meta-arteriole. Now, some of the examples of increasing flow to the capillary bed would be if there's more metabolites. So more of those buildup of CO2, other gases, other metabolites will signal to the precapillary sphincter that we need more flow in order to clear that and bring more oxygen in. So that is an example of causing relaxation because of additional metabolites that will increase flow into the capillary bed.